Hello, my name is Anna Crabtree and I am a double major in Environmental Studies and Mechanical Engineering. This year, I work at the Ohio University Voinovich School with Dr. Miller as a PACE lab assistant. My study is an extension of a previous study evaluating the effects of septage fertilizer on crops. My results will help determine the efficacy of applying septage fertilizer to the plant Agave americana in place of synthetic fertilizer.
Synthetic fertilizers are commonly used to supply crops with nutrients, but excessive use leads to greenhouse gas emissions and environmental pollution via eutrophication. Organic fertilizers are an environmentally friendly alternative that can reduce these negative environmental effects. Septage is a waste product that can be treated using hydrothermal carbonization, referred to as hydrochar, to generate a pathogen-free, nutrient-rich organic fertilizer. To optimize fertilizer use, it is crucial to understand its influence on different species of plants. While organic hydrochar fertilizers have been tested on agricultural crops, few studies have looked into their impact on Agave americana. I am looking into Agave americana because its high potential biomass production in semi-arid regions has a lot of potential to be used for bioenergy, which is a promising renewable energy source that can reduce the need for fossil fuels. 30 Agave americana plants were assigned to 3 treatment groups: hydrochar, the organic septage fertilizer, urea, the synthetic nitrogen fertilizer, and a control, with 10 replicates in each group. Fertilizers were applied at 100 kg per hectare in each plant pot. At the beginning of my study, I recorded biometric data of each plant, including number of leaves and pups, and lengths of the longest leaf, tallest leaf, and center spike. I also measured the soil nitrogen content by sampling soil from each plant. I used an elemental analyzer to determine the carbon and nitrogen content of the soil samples. Through a Spearman correlation matrix of these 2 data sets, I determined the most statistically significant variables. Through running ANOVA and Tukey's HSD tests on these variables, I was able to determine several key pieces of information. These results indicate that there is not a significant improvement with hydrochar fertilizer compared to synthetic fertilizer, but the septage hydrochar can produce similar results for plant growth. This analysis does not yet include the nitrogen and carbon content of the plant leaves, which should provide more insight into the differences between the urea and hydrochar fertilized plants.
The next steps of my study are to measure the leaf nitrogen and carbon content previously mentioned, and measure photosynthetic rates and biomass yields in May when the plants are no longer in a dormant period. Once I finish conducting these experiments, I will perform more statistical analysis to determine how effective septage hydrochar fertilizer is compared to synthetic nitrogen fertilizer for Agave americana.